Hello. So, we were still with that type 3 copper proteins. So, which we know already that they are electron transfer and sometimes they function as some particular part in a typical electron transfer chain. So, if we have an electron transfer chain and type 3 copper site can be involved there. But when we talk about the enzymatic activity like that of tyrosinase activity, that means we want to oxidize the tyrosine function, and in our last class what we have seen that the typical alignment of the dioxygen molecule between the two copper centers. So, this is definitely a dinuclear system, and sometimes if you have the close proximity of two copper centers, we will find that they are close to each other. But if you have the typical dioxygen binding that we all know from the knowledge of hemocyanin chemistry, and we have three other binding positions from the nitrogen residues of the protein chain, that means they are all of nitrogen from the histidine chain, from histidine origin. All histidine origin. So, this we all know from in the case of hemocyanin also, but what is the basic difference if we talk about in terms of its tyrosinase activity? That means the question also will come to us that whether certain assembly where you have a binucleating system and that binucleating system can take up two of the metal centers such that the two copper centers and you get a binuclear copper complex, and that binuclear copper complex is utilized for activation of this dioxygen. And the nature of this dioxygen molecule, because this is typically coming from the O2, the typical dioxygen molecule, it can be your O2 from the air or it can be your O2 from some other source. And around these, that means when both the copper centers are pentacoordinated in type, and this particular histidine nitrogen plays some important role that we have seen that if we have these two copper straightaway we write this function and we bring the tyrosine function, that means the phenol unit, and that phenol unit when it is coming close to one of the copper center, it is not that it is symmetrically positioning between the two copper center, but it is close to one copper center, and the alignment of this is important, this imidazole ring, and the alignment of this imidazole ring will tell us that how it can be helpful for positioning of your substrate molecule. So, the tyrosine end is coming, and there we have seen that it can show when they are close to each other, they can show pi-pi interactions. So, this pi-pi interaction is therefore typically important, because you see that this is some kind of secondary interaction, like sometime we find in case of hydrogen bonding interactions also, that if you have a metal center which is bound to some water molecule or any other function which can donate your hydrogen bond, so that water molecule can show some hydrogen bonding interactions to something else which is not bound to the metal center. Here also this pi-pi interaction, why we want to know all these informations, because for the supramolecular interactions, what we call beyond the molecule, when we have this ligand system, that means you have the 3 donor groups here and 3 donor groups here, and we know that this in the hemocyanin case, when 3 of these, that means the apoenzyme, when copper centers were not there, and initially both the coppers attached to these 2 sites are in cuprous state, is copper 1. Then it goes to cupric state with the transfer of electrons to dioxygen molecule, slowly it is becoming peroxide part. But when this particular part, so it is not that it is one part of the long chain, the protein chain. So this particular part when interacting, and the typical positioning of this will give you some pi-pi interaction, and it brings the substrate near to one of the copper center, which is your copper A. This is also copper A and this is also copper B. Then what happens, this particular positioning will tell us something, that this particular substrate molecule, the tyrosine molecule, with its one oxygen which is close to this copper 1, will start interacting with this copper center. That means it will start interacting with copper 1, and due to that what you have, the still these groups are there. So this dioxygen molecule, this particular moiety which is already bound, if it is a typical dioxygen molecule, you will have a dumbbell shaped structure, that means you have O, this sort of thing, and this positioning of this OO, that means the typical dumbbell shape, this particular type of binding is unique. So not that all peroxo linkage bind in this fashion. Sometimes it binds to this copper and this oxygen and this oxygen, so both the oxygens on one side. So this is the typical binding. So if there is any such interactions coming from these, because the ultimate goal what we are just looking for is that this oxygen is our reactive reagent now, which will attack your ring only, this tyrosine ring, for the hydroxylation reaction. So this the entire this dioxygen group, basically that means this OO moiety then rotates. So the rotation of these two, that means your OO group, that means OO function or OO moiety we basically write, rotates. Why it is rotating? Because we have started something, that means this oxygen is starting its interaction with this copper, and copper will have always a typical preference for a 5 coordinated site, and we are still expanding to go to 6 coordinated site, but that is also a favorable one. But your bonding pattern will change, because there will be steric congestion. So this dioxygen moiety basically rotates from some orientation, we call it as from horizontal to a vertical orientation. That means orientation basically changes, and we sometimes call it that the movement is going from a horizontal position to a vertical position, because these are all 3-dimensional structures. It is very difficult to visualize on a plane of paper that how it is moving, but it is moving in above the plane and the below the plane. So some orientation is taking place, and why this orientation, and we sometimes we put 2 arrow for that there will be some orientation, that means the reorganization of the dioxygen molecule which is still bound to your dioxygen center, and that is so happened that your phenyl ring, that means this part basically is coming close to this oxygen. Initially you have pi-pi interaction for suitable positioning of this tyrosine residue to that of your imidazole ring, and then once that particular positioning is established, that we know that for a typical pi-pi interactions we always have some distance, the centroid to centroid distance, and the pi stacking also we call. So, if this particular distance, which is also within 3.5 to 4 angstrom, and pi stacking is favorable, that means your substrate is getting positioned. So, you see that is a very interesting thing, that your this particular part, that means the ligand part from the long protein chain, which is the histidine residue, which is attached to your copper center, then it is basically bringing the substrate molecule. So, through this pi interaction, your substrate molecule is coming close to this particular entity. It is not that this particular phenol ring is coming and directly attaching to the copper system. What we see in our laboratory experience, that what you get, that if you react with something, that means you want to have something where you can have some ligand exchange reaction. That means, if the phenol is strongly bound to the copper center, your one of the bond for this will go. So, either your entire dioxygen molecule will go or the peroxide linkage will go, and if it is directly bound to this particular one. That is why in biology all these weak interactions are so important, and weak interactions are predominating for their catalytic activity. So, this pi interaction first, then the typical orientation because of this interaction also. When we write in this fashion, that means this bond is bigger than copper oxygen bond. This bond, what we know that typically for a apical coordination, if we find that is the basal plane is there and little bit of apical coordination is a long one, and you have some Jahn-Teller distortion also you expect. Through that you get something that you have a copper oxygen long bond. So, this is also a weak interaction like that of your pi interaction. So, this movement basically bringing your this particular moiety, that means this phenol group, to a suitable position such that your oxygen will be attached to that ring. Still we will consider as the dotted line, because that distance can change little bit. Initially this distance is bigger and this distance is still bigger. It is not within the bond distance, which should be very much similar to that of other known or reported copper oxygen bonds. So, still you have the 3 other bonds from the nitrogens, and this oxygen in the same way you just have with that copper, one bond and one third bond. So, this one then in the next step, this will react with this oxygen. So, your reagent is ready. So, your reagent will transfer this oxygen to this phenyl ring. So, electrophilic attack of this OO moiety, the entire OO moiety, this OO moiety is attacking on the aromatic ring. So, you have an electrophilic attack on the aromatic ring. So, this particular unit though is attacking your aromatic ring, and something else is also happening at the same time. It is the cleavage of your OO bond. This is also important. So, attack of this OO moiety to the aromatic ring and cleavage of the OO bond. Cleavage of OO bond. So, that basically gives us something which is also very interesting to know there, that once it is cleaved, you have the first copper, which is copper A, and you have copper B, and this one sometimes we can write the solid line bond, or you still we can write it as dotted one. This oxygen is now transferred to your aromatic ring, that means your phenyl ring, and this will remain as O, and if you have this thing, that means you have this CH bond here. So, this proton from here will transfer to this oxygen. So, basically what people have characterized, because you never know which particular species you are going to characterize for all these biological systems, and identification of this particular part, that means this copper part as a type 3 unit is very important. That means you can control over here, that means you know that what should be your copper distance, whether it is 3.3 or 3.4 or 3.2 angstrom. So, it is close to say 3.4 angstrom, and that particular one is also not only dependent on the other groups which are bound to these two copper sides, but also the main binding group, the main bridging group. Here this is a main bridging group also, this what you have generated, the hydroxide bridging. So, this particular entity, that means the core structure, you can call it, this is a core structure of type 3 copper proteins, where you know that it has a copper distance of this much, and this hydroxide function is attaching to one copper. And not only this hydroxide function, we also find something, that this phenol unit you have, and this phenol group we all know when it is there, if it is not a terminal one, this phenol unit can bridge to copper sides. But this is not a typical phenol one, but it is a catechol. So, binding of catechol of this sort is very important. So, we are just from the tyrosine we are producing this catechol, and this catechol is ultimately bound to this dicopper center in a fashion. So, binding mode is a bridging mode, and one of the oxygen is utilized to bind both the copper sides. So, is eta 2, and next one is only one copper. So, is a mu eta 2 eta 1 binding mode of this catechol unit. So, that is why we get that the studies of these sort of systems are very important. That means, whenever we get that corresponding model studies or small molecule modeling in the laboratory, immediately we always tempted to get some species where the phenol is utilized as your ligand system. And the only phenol, if it is a terminal one, it cannot bridge the two copper sides. So, if you have certain groups over here like this catechol unit and another group on that side, it would be a very good ligand system which can bind both the two copper sides. And this particular hydroxide function which we have generated from this dioxygen. So, your dioxygen is your source for this hydroxide function. But sometime when we go for some laboratory experiments, when we do something in the laboratory, we occasionally find that this is not coming, if it is not going through some redox reaction, it is not coming from O2, but the major source for these hydroxide bridged dicopper complexes are water molecules. Because immediately if you see that water is bound, sometimes water is bridging the copper system, and when both the two copper centers are bridging this or sharing the lone pair of electrons on the water molecule, you have immediate deprotonation by changing the pK of the water molecule and you get hydroxide of bridging. So, once it is happening, that means you have these two copper sites which is hydroxide of bridging, and ultimately what we get, we just then the release of this product in a form which is not the catechol bound form. What is released from there is the oxidized version of the catechol. So, it is the quinone which will be released from there. So, that quinone, so it is the oxidized form, and because what is happening, everything is happening simultaneously together. That means, you have the hydroxo bound system, and this particular catechol unit is bound to the dicopper unit as mu eta 2 eta 1 fashion, and that eta 2 eta 1 group when it is there, and this 2 copper is bound, because initially when we started from here is that we get that thing that you have 1, then electrons are transferred to this dioxygen molecule, you have the copper 2. So, these are copper 2 before the release of the product. So, immediately what we find that the catechol unit is then responsible for the reduction of the copper centers, and your catechol unit is getting oxidized to the quinone product. So, you have the quinone product which is liberated. So, it is not for any one such quinone product. So, all sorts of reactions having anything, that means having a tyrosine part, because we are talking about something which is amino acid based thing. So, any of this type where you have some pendant tyrosine unit, any protein chain having a substrate unit, that means TYR is there, that means one part is tyrosine and the pendant tyrosine OH is involved, or the O group is involved for copper coordination, there will be a possibility for going for this corresponding oxidation in that particular part. So, for this all the hormonal molecule and all these things where you have the catechol part is involved, initially if you do not have any such oxygen, you get it from the hydroxylation reaction followed by the tyrosinase activity. So, the tyrosinase activity is responsible for that, and this OH group will be taken care by the H plus available in the system, and it will be released as water. So, you will end up with the system where you have the corresponding deoxy form of this copper. So, this deoxy form of these copper centers, basically then it will be released as one copper and another copper without any bound dioxygen unit over there. So, both these deoxy form, that means this particular deoxy form you get and your quinone product. So, quinone product is liberated or released. So, that gives us some clear idea about all these groups, what is being involved for this dicopper chemistry. Basically, at the same time we are learning so much thing about the dicopper chemistry, and if it is OH group which is either forming from the dioxygen molecule or it is coming from the water molecule, we can go for something, that means how we can spectroscopically characterize. That means we are little bit we are going back after knowing the mechanistic path for this transfer. So, if we have any such system where you have the copper 1 which is bound by OH group, this is important. So, in that particular case we get also another bridging from the phenol unit. Basically, what we get a unit which is known as a Cu2 O2 unit or a Cu2 O2 core, and when this Cu, this is one oxygen of say phenol and this is another oxygen from hydroxide O unit. So, you get a typical diamond like unit. So, Cu2O2 units, this diamond like unit, because this is very important to know, because structurally we can know so many things out of this. Because if you just compare this particular part, that if you have this, why people have very easily identified this basic unit of that, instead of having a type of interaction like this from a dioxygen molecule. This is one of the possibility. So, when this dioxygen, both the oxygens are not attached to the copper. So, one of the mode of peroxide binding is this binding, and 3 dimensionally when they are basically rotating in 3 dimension, what we find that this sort of oxygen bridging, these are the bridging units. So, bridging oxygen atoms you have, and if we just can determine, this is a very simple unit, but structurally if we can see that there are 4 copper oxygen bonds, and we know the regular bond distance depending upon the nature of the oxygen, whether it is water oxygen or hydroxide oxygen or oxo oxygen or phenol oxygen, and copper in the corresponding oxidation states, that means is a Q plus copper or cubic copper, you can have these copper oxygen bonds. Then if you know these corresponding bonds and then you try to compare this, immediately you will find that if this oxygen, when they are making bonds with this copper, this particular copper oxygen bond and this particular copper oxygen may not be equal to each other. That means one of these copper oxygen bond can be short and other one is long. Similar thing also happens to this oxygen and this copper. That means it immediately tells you that whatever we are drawing on this plane of paper or the piece of paper, and we are talking that this is a diamond core, but what we are drawing is in a plane of paper. That means we are restricting our drawing on the plane, and we are thinking that if all the 4 atoms are on the plane of the paper, but it is not that. So, if this oxygen is going up or going down, one is going up, another is going down. So, what basically we get when we think of that, you have a symmetrical binding of from these oxygens, either it is from the hydroxide oxygen or phenol oxygen. That means if they are all in plane, it can be a symmetric one, otherwise you can have a long bond and you can have a short bond. Why this is so? Because if you just think that copper, this particular copper is in square pyramidal geometry, or the other alternative is your trigonal bipyramidal geometry. So, now think of it that you have a 3-dimensional structure and your life is much more complicated now. So, these 2 copper, this particular basic core, whatever we are talking about, we are thinking that is in the plane of the paper, but once you draw the other 3 bonds, this bond and this bond, now is a complicated one, whether you have a copper center of this or a copper center of that. And there also we get something that you have these 4 bonds when you have a square pyramidal geometry, these 4 bonds are short compared to this apical bond. So, we are talking something, these bonds we call as the basal bonds and these we call as apical bonds. And all these copper oxygens, that means the copper oxygen apical bond distance will be greater than copper oxygen basal. Similarly, in this particular case also, this is square trigonal bipyramidal also, you have one basal, one apical. So, initially, because mechanistically also you can think of, you can just think for a while for this, that whenever this oxygen is coming into the picture, starting slow interaction with the copper center, and then it is forming the bonds. Not that all the bonds are forming together. So, one is approaching first, then it goes for the second bond. Initially, you can have this sort of interaction. This immediately tells you, if you draw instead of drawing this, if you draw in this fashion, that immediately will tell you that this solid line is the shortest line and these are the long one. So, all the time you get that. So, you have some asymmetric geometry. So, this asymmetric geometry will tell you, and if we want to characterize this, because particularly this type 3 copper is a basic component from some other particular proteins which we call as multicopper proteins or multicopper oxidases. These are multicopper proteins and they are multicopper oxidases, we sometimes we call them, such that this ascorbic acid oxidase, ascorbate oxidase and all. So, when you label it as a multi-copper system, that means you can have more than one copper center. So, the minimum one, the binucleating one, how you characterize it first. So, already we have seen the type 1, the T1 center already we have seen, then T2 center type 2 proteins, and now we are in T3. This multicopper oxidases are sometimes is such that you can have all the 3 types together. You see the situation is getting much more and more complicated. So, individually if you are able to characterize the copper 1 site, all the typical spectroscopic properties and all other physical characterization detail if you know, you can find out T1, you can find out T2 or you can find out T3. And while characterizing this thing, that means they are showing this oxidase activity. So, you have a system where you have more than one copper center is there. Sometimes if you have a system, that means all 3 are present together, the type 1 copper, type 2 copper and type 3 copper. That means you are having a system where you have 4 copper centers, which will be a tetranuclear one. But in the biological system, what people have tried for that, and we succeed to characterize it, that if it is not a different one, that means it also contains a T1, that means the typical signatures for the T1 is present for this, T2 is there and T3 is there, and if you sum up all 3 types, it will be 1, 2 and plus 2. So, 4 copper centers will be there. And when this particular multicopper oxidase is showing the oxidase activity, and initially when we try to know the oxidation state, that means whether these 2 sites are copper 2 plus or one of them is copper 2 plus, one is copper 1 plus, or both of them are in copper 1 plus. That is also important, because every time we are looking for something where electron transfer taking place and they are basically reversible one. What you can have from a reduced species to a oxidized species, you can get several E0 or E half values, which will be characteristic for a single electron transfer. That means one electron you are putting, the second electron you are putting, or one electron you are taking out, or the second electron you are taking out. So, one of the characterization value for all this center is your determination of the redox potential value. So, redox potential you determine first, and then the corresponding distance, that how far these 2 copper sites are. If they are bound by a hydroxide group, then the resonance spectroscopic technique, that means the due to unpaired electron, that means we can again utilize the EPR spectra. So, that EPR spectra is related to that you have a corresponding unpaired electron. Then if you have a unpaired electron system, and if you have one unpaired electron when both of the copper centers are +2, one unpaired electron, one first copper, that means the copper A, and the second is copper 2, and if they are showing some signature that the center, that means the type 3 center is EPR active, that means you have some unpaired electron present over there. So, some amount of unpaired electron density is there, that means your both the two unpaired electrons on copper A and copper B are not interacting very strongly, such that your ground state is S is equal to 0. When it is S is equal to 0, that means it is paired up, you will not find any EPR activity. Otherwise, you can have the corresponding determination for the J value, the coupling constant. When your magnetic moment is less than that of your sum of the individual centers, we call the J is there and they are engaged in some anti-ferromagnetic interaction. Why we are determining this magnetic property? We are talking somewhere the catalytic behavior. Now we are talking the anti-ferromagnetic or magnetic part, because this magnetic characterization is very important, whether in all sorts of things, whether it is a copper based system or a nickel based system or a cobalt based system, those who are the metal centers are providing unpaired electron, whether there is some relationship between this magnetic property, because it is a physically measurable quantity. So, you have now some very sophisticated magnetic instruments or magnetic technique is available. In older days, say 50 or 60 or 70 years back, we are used to get this magnetic moment, the simple mu b, by using a balance. So, balance you have, then you take the two different weights in the presence of a magnetic field and in absence of a magnetic field by a typical Gauss balance technique. So, this particular mu b, the magnetic moment when you get, that means how much your electron density is residing on these two copper centers, that means how they are interacting to each other. So, the individual orbitals, the atomic orbitals which were present on the two copper sides and the unpaired electron on these, that means the dx square minus y square or dz square, how they are interacting is depending on this picture. That means what 3-dimensional structure you have, that means either you have a square pyramidal structure or a trigonal bipyramidal structure. That structure will tell you which orbital will be available for your unpaired electron, and then how close they are, that means this distance, this copper-copper distance, which is also important, how close they are for their suitable interaction, because everything what we are getting over here is not that based on the carbon geometry which can go up to attaching to force some species on the carbon center, but this when it is attaching to 5 such species, and these orbitals are very important and these interactions. So, magnetic moment you can correlate with those groups, because in these multicopper oxidases, when we have more than those groups, say for ascorbate oxidase, one is ascorbate oxidase, and sometimes we can have also the ceruloplasmin, or sometime Lec-13. So, this particular part, that means how the same copper site which is being utilized for tyrosinase activity, which can be utilized for ascorbic acid oxidation also. So, we know that this ascorbic acid group is a very good reducing agent and which can be utilized for your ascorbate oxidation. So, when it is in the deprotonated form, we call it as ascorbate oxidase, and otherwise we typically call it as a corresponding oxidation reaction for your ascorbic acid. So, it is not a very simple system, because it is a multicopper oxidase molecule. So, you have more than 2, 3 or 4 copper centers present over there, and when we characterize, that means we will find that it has a color which is very much similar to the one type what we have characterized earlier, which is our type 1 type. So, type 1 copper center is present. So, if we get something, that means the band is close to 600 nanometer or say 620 nanometer. So, that is the corresponding signature from the electronic spectroscopy. So, that electronic spectroscopy immediately tells you that these multicopper oxidases can have a blue color also, because you have a corresponding band at this particular nanometer, and it catalyzes the oxidation of ascorbic acid. So, is the corresponding oxidation of the ascorbic acid. So, this is our ascorbic acid. You have so many OH groups, and during deprotonation also we should know that which particular OH group is getting deprotonated such that it is producing the corresponding ascorbate anion, and this basically is reacting with half of this O2 molecule, and your ascorbate oxidase or ascorbic acid oxidase, sometimes we abbreviate quickly as AAO, which is ascorbic acid oxidase. So, ascorbic acid oxidase is there. So, when the color is there, that means you have at least one type 1 copper center is present, and we call it as also as a blue oxidase due to its color. So, it is a multicopper oxidase which is also a blue copper oxidase, because it has color and it has a type 1 copper center present. So, it is getting oxidized simply like your catechol to quinone conversion. So, catechol to quinone conversion is taking place over here again. So, if you know nicely the tyrosinase activity or catechol oxidase activity, you should be able to tell what should be the product for this reaction. So, this is also one H is also you can show, because all 4 bonds we are showing CH to OH, this is HOH. So, this is our ascorbic acid and this is our dihydroascorbic acid, and which is producing also one molecule of water. So, whatever oxygen we are supplying to the system, that means this half O2 azo O2 which is getting utilized for H2O. So, it is a typical oxidation reaction what we get like your catechol to quinone conversion. And similarity why we need that copper there. So, you have 2 successive 1 electron steps. So, how you get that is that it means the how this oxidation step is taking place, because we are thinking that copper is there. So, the involvement of the copper. So, if you have a phenol unit, if you have a tyrosinase unit, we know that that particular phenol unit or tyrosinase unit is responsible for copper coordination or copper binding. So, this coordination is important. So, you have to establish first the coordination to the copper center, then you think of any change in the corresponding oxidation state on this copper, either it is a cuprous copper or a cupric copper. Then you bring your reagent, the dioxygen molecule. So, how all these things are taking place there. So, this particular unit, when we talk in terms of that ascorbic acid oxidase or ascorbate oxidase, that means it can go for deprotonation nicely. So, this deprotonation will give, so now onwards basically, because getting this particular part, because this is the part which we are interested to know, because this particular part is the catechol part. So, the cis dihydroxy groups are there and those groups are basically utilized for copper binding. So, this particular hydrogen is going away. So, this one, once it is formed there, will start interacting. So, one in OH form, another is O minus form, will start interacting with the copper, and that particular copper, what is present over there is your copper center, and this is the copper 2 plus 2 center, and it belongs to the enzymatic part. So, your entire enzymatic part and is a very complex system where you have more than one copper center, and this particular one is bound and this copper is going over there. And then if you think that this dioxygen is coming into the picture, that means what we have this copper, and then copper is responsible for this dioxygen binding, what we know already. So, this particular part is behaving as your bidentate ligand part, and that bidentate ligand part is attaching to the copper center, which is already bound to the enzyme part, and that particular copper center or the second one, the other copper center, is responsible for dioxygen binding, basically giving rise to something where you get a radical product is the corresponding oxidized form, and copper is reduced to copper 1 and still bound to your enzymatic form. So, this we can study. So, how this copper bound to this enzyme, that means this copper metalloenzyme is attacking on the ascorbic acid or the ascorbate anion. And if we can look at something that what is our product, because in some cases we will find that these products, because the fate of this dioxygen molecule can either be water molecule or it can go to produce hydrogen peroxide. So, depending upon that, and in this entire reaction what is there, and this formation of this free radical can be proved if you just record this particular technique is well known to us now, the galactose oxidase and all these things. Whenever you have the suspect that no, I am not getting something where the copper oxidation state is typically changing, your substrate or the particular part is getting oxidized. So, if you in a transient form also, because the lifetime of these free radicals, if it is very less, sometimes some solvents are available, and those solvents basically we get because the DPPH type of thing, which is the standard reference molecule for recording your EPR spectra. So, some solvent mixture is there, acetonitrile or dichloromethane or some mixed solvent like acetonitrile benzene or acetonitrile toluene. Those solvents can be frozen at a temperature of 77 K, which is our liquid nitrogen temperature. So, at this temperature you can freeze the entire system, and the frozen system can be inserted within the EPR spectrometer probe, and we record, that means whatever lifetime you have is a transient lifetime it will have, but you can freeze that free radical and that free radical can give you the corresponding signal which is due to your presence of free radical. Okay. So, this multi-copper system what is present and the entire reaction, that means how many electron transfer basically you can expect if you think that in this particular case you have generated the copper 1 system. So, altogether when you reoxidize the system, this is the reduced form of the copper enzyme which is copper 1 bound to the enzyme part. When handling our O2, we know that what we can have 4 electron transfer like cytochrome C oxidase. Still the same thing we are doing, we are handling on the dioxygen molecule, and that dioxygen molecule with 4 protons on it, it can go for 4 copper center in +2. That means, when you regenerate the enzyme system in the cupric form plus 2 H2O. So, the dioxygen there, that means this particular case. So, when the enzymatic function, the dioxygen can be utilized to generate the copper in the +2 oxidation state. That means, you are just segregating the part, that means your substrate is here, your copper 2 is there and enzymatic function is there. So, if you just think of the corresponding electron transfer between your ligand and the metal center, you get the oxidized ligand version and the reduced metal center, but we are not talking anything related to the electron transfer to the dioxygen. Now, the reduced form of the enzyme, is then finally reacting with the dioxygen molecule to regenerate the cupric form. So, how we can think of this? That means, this basically a system where you have one particular species is this is another and this is another, that means the involvement of the proton, and when you have this copper as one part, enzyme as one part and the dioxygen molecule is one part. So, you have a 3 component system. So, when we go to the laboratory to study this system on simple model compounds, so when we think something on the model compounds, that the model systems for model study, and that model study we can make with simple copper 2 ion or any copper 2 complex. That will immediately tells us that what is the difference between the simple copper ion and a copper present within the complex. Because this particular part, that complex part, what we are getting this particular part giving you the ligand system. So, it is the enzymatic part which is basically modulating or controlling the corresponding electronic behavior of the copper to be reacting with the substrate molecule. So, if these two, that means the copper 2 plus and the copper 2 complex are reacting with this dioxygen molecule, and if we are able to monitor the product of these reactions, that is one very simple clue to know a critical mechanistic pathway for any reaction, forget about for this ascorbate oxidase or anything. Whenever we talk about something related to a metalloenzyme, how you know the corresponding enzymatic function, what we want to establish that it has some role, that means copper is playing some important role, and what is the role of the ligand part. So, you just differentiate these two, that means the copper 2 ions, and in most of the cases if this particular reaction is producing immediately the hydrogen peroxide and this reaction is producing water molecule, definitely you will tell that the course of the reactions are different. And in case of this ascorbate oxidase, your product is not hydrogen peroxide, your product is your water molecule. So, this particular one, that means the copper, you have one copper center, then one of the ligand center or sorry the substrate center and the dioxygen center. So, you have the ascorbate anion, then copper and then dioxygen. So, it is basically a system what we commonly call as a ternary complex. So, when you have the ternary complex, we get some rate expression for the rate of the reaction, and rate expression we see that basically if we write the rate expression, the dependence, that means it is dependent on the concentration of say H minus, that means the ascorbate anion. It is also dependent on the O2 concentration and it is also dependent on the copper 2 plus concentration. So, what we get therefore, that whenever we have certain these reactions, that means based on the ligand part which is our substrate now, this is basically reacting with your copper, we have the copper and then the dioxygen. This is the system. It is a very simple system that you have substrate which is functioning as a ligand. So, this particular entity you can consider as a metal complex, the substrate plus metal center, then it is reacting with your dioxygen molecule. And this particular one, when it is copper 2 plus and this one when it is interacting with this copper and this dioxygen, and you just go for this and this is going for that corresponding dihydroascorbic acid formation, and you can judge the radical formation and ultimately you can see that how the electron flow is taking place through this oxygen. So, ultimately you are moving from here to there. So, that is the thing, that how this electron flow is taking place via this oxygen. Okay, thank you.